The next speaker, and after that we will have a break for lunch, and then we reassemble to complete other presentations, is Professor Bernard Rodriugues, who is working for several years in the Department of Botany on a very important economic industrial aspect of beneficial fungi, that is mycorrhizae. Now mycorrhizae are eco-friendly associations of plants with fungi. There is hardly any plant which does not have a mycorrhizal association. Dr. Bernard has painstakingly created facilities which you can visit separately and see what he is doing in various glass houses, greenhouses, hothouses and all. So I request here Professor Rodriugues to present the world of mycorrhizae for you, which is going to get more and more popular, where people are looking for carbon credits, eco-friendly technologies. So for media, there would be a lot of interest in the coming years in this technology.
Well, good afternoon. The earlier speakers, they talked about terrestrial and aquatic habitats, the diversity or the biodiversity, whereas let us for a while see the amount of diversity that exists in the soil. Soil is an important abiotic component which harbors lots of living organisms, microorganisms, and these microorganisms play a very important role. Just the other day we were discussing that if these microorganisms are not there in the soil, what would happen to our soil? And if the soils are inert, lifeless, what would happen to our vegetation? And then, the human race would be extinct. Well, I'll be talking on a group of organisms, just one group of organisms which are very important. These are mycorrhizae, and their role and how we are conserving mycorrhizae in a little possible, effort from our side at the university. Next one. Yeah. The mycorrhizal system, it consists of the plants, the soil, and these fungi. So these fungi, they are obligate symbiont, that means they cannot do without a plant root, and also they are useful for the plants. And most of the plants that we have on this planet, they are mycorrhizal. More than 90% of the plants are mycorrhizal. Next one. Yeah, they play a very important role in the bidirectional flux. That means the fungus would provide phosphorus to the plant, available phosphorus, would convert phosphorus, all the non-available forms into available form and make it available to the plant. And in turn, the plants would supply carbon compounds to the fungus. Thereby, it's a symbiotic association. You can see here the plants, the first figure on the left is the plant with the plant roots. And these roots, there is attachment of this, or there's a association of these fungal hyphae which are in white. And as a result, these fungal hyphae, they'll get into the soil at a zone, at a region beyond the root depletion zone, and would absorb the phosphorus and make it available to the plant. And that's how there's a plant growth. Yeah, apart from this phosphorus, there are different roles which are benefits of these arbuscular mycorrhizal fungi to plants. Next. A simple experiment how a mycorrhizal and the non-mycorrhizal plants behave. When a mycorrhizal inoculum is added in the pots, you can see that there's an increase in growth, and there's about 2 to 2.5 fold increase in the plant dry weight, as well as the yield is more in mycorrhizal plants. Next. Now when we talk about these mycorrhizal fungi, they form peculiar structures which are found in the plant roots as well as in the soil. You can see on the left, these are the arbuscles which help in the nutrient transfer to the plant. Then these are the second is the vesicles. They are round or ovoid structures which act as storage organs. They store oil droplets. And you find those two structures, the third and the fourth, these are the auxiliary cells which are present both in the soil and in the plant roots. And also on top, you can see the colonization by these fungi. You can see a lot of hyphae with a few vesicles. Next. Yeah, what we do is we try to isolate these spores from the soil. There's a good amount of spore diversity in the soil, And these spores have to be isolated before we prepare cultures out of from the spores. So by using this wet sieving decanting technique, we isolate the spores. Next. And then we maintain these trap cultures, wherein we use the spores, trap them, and then make them to colonize with the test plants such as Coleus species, or for example, chilies or onions, and then multiply these spores, because these spores would germinate in the pot, they would colonize the roots of the plants, and as a result, they would multiply and sporulate. So as a result of sporulation, the number of the spores would be more. Next. You can see the spores that are isolated from the trap cultures, where there are large number of different species, and below you can see the spores which have been recovered from a pure culture. So we maintain these pure cultures in the polyhouse, which is totally dedicated for the maintenance of the pure cultures, and these pure cultures are later on used for experiments to study the growth and the yield in different plants. Next. Yeah, coming to the EM fungal diversity, there's a good amount of diversity, and we have recorded about over 80 species of arbuscular mycorrhizal fungi from different habitats. See the mangroves, the sand dunes, then mining, and various other habitats. And they vary in color, shape, size, ornamentation, the wall layers, all these things they vary. And these have to be maintained separately in the pure cultures. Yeah, this is another group of species. Now, what has happened is they are obligate symbionts. That means they need a living plant root. And also there are few problems when you maintain them in pot cultures. The conditions have to be, very clean, and there should not be any contamination. And what the recent techniques, that's about 20 or 30 years old, where people have been working on in vitro cultivation of these organisms. So the spores are grown in culture media with special media, wherein the spores would germinate and colonize the roots. So that is the in vitro technique that we have been following recently, and we have achieved a good amount of success as far as cultivation of these spores or the species is concerned. Next. That is the process how the in vitro cultivation. Next. Yeah, these are the roots of plant species, selected plant species, which has been transformed and which are used for multiplying the mycorrhizal species. Next. You can see the spores which are put inoculated in the presence of the roots which are transformed. Next one. We also make these the spores to germinate by using artificial media, so that the spores would germinate. They'll produce those germ tubes, and again, next. Yeah, these are some other species wherein they are germinating. Next. Again, the spores which are germinating different species. Next. This is another species where it is germinating, and the germ tubes have. Yeah, just before that. Yeah. Yeah. Then once the spores germinate, we transfer them into the plates containing the roots. And these hyphae, they would colonize the roots. Next. Yeah, the next. And once the hyphae, they colonize the roots would also grow because they are transformed roots. And as a result, at the end of, say, about 1.5 to 2 months' time, you find there will be a lot of sporulation. A large number of spores are formed in the petri plates, that is in vitro. And as many as 5,000 spores can be produced in a single petri plate. And this could be used as an inoculum. By using a suitable carrier, we can use it as an inoculum for inoculating the plants. Quite a number of spores have been obtained in pure cultures. These are some of the pure cultures that we are maintaining at the Goa University. Next. Now, these spores, this inoculum that is produced by in vitro culture techniques as well as the pot cultures could be used for application in different ecosystems or the habitats, such as the sand dunes, which are degraded, so they need attention. Next one. The mine reject. So we have done some amount of work as far as the use of mycorrhizae in the mining is concerned, and we found that mycorrhizae play a very important role for the simple fact that there are no plants which are not mycorrhizal and surviving on the mines. Especially when the fresh mining is done and there are all rejects, whichever plants they invade these mine reject dumps, they are all mycorrhizal, indicating that mycorrhiza plays a very important role in the survival of these plants. Next. Yes, in the nurseries, the mycorrhizal inoculum is used for raising the nurseries and then plantation to the field. Next. In tissue culture plants also, mycorrhizae are used. Next. These are some of the results that as far as the colonization, there is increased amount of colonization by using different species. What I have shown, GI, GM and GI plus GM, these are the different species or a combination of the species. Next. Again, on biomass, you can see the effect of a particular species on biomass. It again varies. Therefore, it is important that we have to identify which particular species would suit for a particular plant. So accordingly, we have to recommend that that species could be used as an inoculum for that particular plant species. Next. Yeah, this is in case of Syzygium, that is jambul. Next. Yeah. So when we use these especially in areas which are degraded, the mycorrhizal inoculum, then we can achieve climax vegetation or maximum diversity in the shortest possible time. Like they talk about if a mining reject or the mine dump is left unattended, it will take about 150 years for it to get naturally colonized by plants to achieve, and one would achieve the climax vegetation. But this can be done within less than 10 to 12 years spent by using mycorrhizal inoculum and by using the plants which are colonized by mycorrhizal. This is a book that we have brought about, that we have published in 2009. It gives a detailed account of the inoculation techniques and the diversity of different species that are found in grower, and it's a manual where the beginners in mycorrhizal research can always refer. So it's a good guide for the beginners. Next. Thank you.